Today, we're lucky to be joined by Dr. Alice Saragni. She's an assistant professor in the David Geffen School of Medicine at UCLA and a member of the Johnson Comprehensive Cancer Center. Her laboratory is in the Department of Orthopedic Surgery and couples basic research into mechanisms of disease to the development of novel therapeutic strategies. Her expertise lies in the development of tumor organoid models to investigate the biology of rare tumors and perform screenings for functional precision medicine applications. In 2020, Dr. Saragni was one of the first speakers in the series, and so today we're bringing her back so we can get an update to see what's happened over the last 3 years. And the title for today is Sarcoma Patient-Derived Organoids for Precision Medicine. Dr. Saragni, I want to turn it over to you, and thank you for joining us today.
Thank you very much. I'm gonna share my screen. Let me know if there is an issue.
I can see it.
Okay, perfect. So yeah, I wanna really thank the Alex and Melissa Foundation for the opportunity to be here again today after almost exactly 3 years. And as Jay just mentioned, I was speaking. I was one of the first speakers of this series. And it was May 2020, and we were home, and it wasn't a great time. And I remember giving this talk as, one of my fondest memories of that time. It was one of the few fun science things I got to do then. And so I'm really excited to be back and tell you how things have evolved over the past few years. And so just to kind of catch everyone up to what we do, we like to develop patient-derived models. We like to do that to study the biology and for, screening purposes, so for precision medicine. And I'm going to tell you a little bit about these later. But just to start, we work with patient-derived cells. And when you do that, you actually don't have a lot of flexibility as to how you grow these in the lab. Solid tumors in particular are very challenging to just put on a plate and grow in 2D without any modification. So that doesn't really work. You can take these cells and put them in a mouse to develop a patient-derived xenograft. There is some brilliant biology done that way. For us, and from the perspective of being able to do large screening, this is obviously very challenging. It may take a very long time to establish patient-derived xenograft. You need to passage them, and as all these things happen, you can end up either selecting or inducing possibly changes which are not reflective of the patient tumor anymore. And also in particular for the type of tumors we study, some of the sarcomas we investigate are very indolent, they're slow growing, they're just really impossible to grow in mouse with very, very low take rates. Now, the easiest thing to do is actually to take these patient cells and put them in a 3D protein matrix, so like an extracellular-like matrix in vitro. And when you do that, you actually are able to grow these what we call tumor organoids with very high success rates. You can get these established very quickly. From unmodified patient cells, you maintain certain components of the microenvironment. They go back restructuring themselves in culture. You have cell-cell components, cell-cell interactions that are maintained. And so these turn out to be really useful models, and they allow us to also perform potentially very large screening to investigate the landscape of drug sensitivity. And while I say they can allow us to do large screening, in reality, when we started this work, we had some really big technical challenges, and screening in 3D was really actually complicated. And it all had to do with the way we typically grow cells in 3D in culture. And what people would just conventionally do is either use U-bottom plates where you have cells kind of clumping together at the center of your well, or you could make a drop of matrigel in a well. You could cover your well entirely in matrigel, which is one of these protein matrix we grow cells in. And what all of these methods have in common is they actually take over the center of your well, which is really challenging then to put together with the type of liquid handler that you use for automating drug screening. So the way in which we went about this, and I hope you can see my mouse, and if not, please say something. The way in which we went about this was to simply reverse the geometry. And so instead of using the center of our well, we ended up using the periphery. So this is one well of a 96 well plate, and you can see your organoids happily growing at the periphery. Center is completely empty. And this allows us to use, any type of liquid handler and automation protocol you may have. We don't need a lot of cells, which allow us not to actually expand these in any way, in vitro or ex vivo. We can, again, really adapt this model to any type of protocol you may want to. The way our usual paradigm works is as follows. We get cells from any type of surgical procedure, really, and we dissociate these down to single cell or small cell suspension. We generate our rings, and then we let the cells, do their thing and grow and expand over the next 3 days. After that, we are ready for drug treatment, which we perform by doing complete media exchange. And on day 6, we can release the organoids, and we can do a final endpoint assay to measure what was the effect of the treatment over the cells. So again, this is very straightforward. And the good thing is, after the first day, we never actually have to touch these cells again. We don't have to move them from plate to plate. We don't have to disrupt them or disturb them in any way. And everything can be done, with robots. Now, as I said, we're going from surgery to possibly identifying an effective therapy within a week. And the reason why we do this so quickly is because we are trying to potentially develop this type of technologies for functional precision medicine. So again, surgery to identifying a therapy within a timeline that's therapeutically and clinically actionable. I think everyone is really familiar with the concept of genomic precision medicine, right? So you take a tumor, you do some sort of sequencing, and you identify an aberration, which is actionable. You can couple that to a specific drug, to a specific intervention, right? And the concept is absolutely brilliant. However, what many trials over the years have shown us is that the results are still not great in practice. And so this is just one, study, the NCI match trial, where patients that had all these different types of alterations were enrolled and given the drug that you see here in this list. Now, the first thing, that you will notice is that the prevalence of this type of actionable alterations is actually very low. That means that only about 30–35% or so of patients are found with with a defect in their tumor, which we can target with a drug today. And also, if you look at the response rates, those have not been great, right? Because you are going in with patients which all have the alteration. You are expecting to respond to the drug, you give them the drug, and then response rates are really low. Depending on the trial and the type of intervention, never really above 30–40% when you do multiple drugs. But for the sake of the conversation, even if all of the patients which are associated, which are given an intervention through genomic precision medicine were cured, you're still left with over 70% of patients for which you have absolutely no available intervention. And so the idea of functional precision medicine is, we don't need to know or have a biomarker to identify an effective drug. We just take the tumor as is, and we test as many drugs as we can to find what may be effective. And we want to do this, in the context of a few different types of interventions, right? We still screen chemotherapy because there are sometimes many options, and also because for some of the rare tumors we are studying, as I'm going to show you in a minute, there is no standard of care. But we also want to do this to screen target therapies. Now, I just showed you even targeted drugs for which there is a biomarker, you still are left with a lot of marker positive patients which end up not responding. On the converse, there is always a component of marker negative patients that may end up responding. And so can we find these different populations? But realistically, the absolute majority of these targeted drugs we develop do not have a biomarker to stratify patients. And so can we apply screening approach like these to find patients and stratify patients for therapy, can we use this as a companion diagnostic to identify patients that are more likely to respond? As well as can we use these, together with next generation sequencing to develop those better biomarkers we so desperately need. We do work on rare cancers. Depending on how you define rare cancers, you can end up accounting for up to a quarter of all diagnoses. This is up to 400,000 people a year diagnosed with a rare cancer. And I know I don't have to say this to this audience, but every pediatric cancer is a rare cancer. Some we know very well. We have good hands-on biology, drivers, therapies, but really for the absolute majority of rare cancers, we don't know much. We have limited sequencing, we don't know about drivers. And as I said, quite a few of them, we have no standard of care. And there are obvious challenges, there are some trials out there where it took a couple of decades to get 100 patients to investigate a first-line therapy. And limited options often for a rare cancer patient to enroll on existing trials. And so again, there is this big window of opportunity with tumors that have no good intervention to apply a technology like this and try to find things that could be investigated clinically. We really study a lot of different rare cancers at this point. We have projects on, the other thing I want to mention is that we actually span the whole spectrum from benign tumors, which have their own sets of issues, all the way to, malignant aggressive cancers. We have projects going from neurofibroma, pheochromocytoma, paragangliomas, etc., all the way to sarcoma. Sarcoma is definitely our largest project at this point. And these are tumors arising from connective tissue, muscle, cartilage, bone. They are incredibly diverse, over 120 subtypes, and this number keeps changing and going up, honestly, and they are all incredibly rare. The way in which our pipeline works is we get tissue from any type of procedure, whether it's a biopsy or a resection on primary tumor, local recurrences, or metastases. We expedite the tissue to the lab where we perform, exactly what I showed you a little bit earlier. We get the tissue, we digest it, we generate our organoid culture, and then we perform characterization. So we do drug screening, we also do sequencing and histopathology. We are about, I believe we are around 170 plus patients now we enrolled in this study, but we have a paper ready to go with the results for the first 127 patients or so. Now, as I mentioned, sarcomas include bone tumors like osteosarcomas, also other tumors like Ewing sarcomas, etc., which are common in the pediatric population. And so about 12 to 15% or so of our patients are pediatric patients. We also have a very large component of young adults, and so 18 years plus. We skew slightly male, and although we have a slight preponderance of white patients, we have a fairly large cohort of patients of Hispanic or Latino origin due to our catchment area. And as you break this down by, the different histologies with studies, you can really pick up on the trends. So you see osteosarcoma really predominant in the pediatric and young population, and young adult population, same for rhabdomyosarcoma, Ewing sarcoma, etc. So we can really look through the data in a lot of details. Now, from these 127 patients, we actually got about 193 samples. And this is due to the fact that we can, for example, get patients that come here for their diagnostic biopsy and then get a resection and then maybe metastatic, and so get successive surgeries to remove metastases. Also, when this disease metastasizes, which is typically to the lung, very often there are, individual metastases within the lung, and we can get these tumors separately to investigate them. So overall, we have screened about half of the under the 93 patients we have. So up to 300, this is 29,000 wells we have looked at so far with very high take rates. Because we are a referral center, a lot of our patients are advanced and so recurrent metastatic, and have seen a lot of previous systemic lines of therapies. And again, as you break it down by disease type, you can kind of pick up on specific trends. For example, you'll see that radiation is given in a high proportion of patients with soft tissue sarcoma, because for these patients that is a very typical treatment modality. How do these sarcoma organoids we keep talking about look like, really? And so they look as diverse as sarcomas are. And that really is the power, I think, of these models. They are very different than carcinomas. So if you are used to the idea of an organoid being just a ball of cells, as carcinomas are, sarcomas are not like that. But sarcoma are not like that in the patients either. And so, for example, you have sarcomas where you have, like this is a rhabdomyosarcoma, where you have scattered cells within a matrix. And this is exactly what we want to develop. We want to be able to maintain the histology and the histopathology of the tumor of origin. You have tumors which are more dense. This is, small round blue cells for Ewing, and that's exactly what we also get in our models. I did mention, these endpoint assays we do, but we also can monitor cells growth over time using a bespoke machine learning based pipeline we have in the lab that allows us to determine their growth pattern ex vivo. We can, of course, then test drug responses. We can do single agent, we can do 2 to 3, different agent combinations and differentiate responses. This is just, some individual examples. For example, pazopanib in primary osteosarcoma, and you see very differential responses. Mosetinostat and vinorelbine, which has now been looked at clinically for rhabdomyosarcoma. Again, very, different responses between different patients. The other thing we can do at this point, because we have screened so many samples, is we can start to look for trends, right? Do we see any correlate of response with any of the clinical characteristics or any other type of feature of the tumors? And so there are some trends that are emerging, and we are measuring these. And I don't think we have yet a biological explanation as to why we're seeing some of these things, but this is what we can now go back and investigate in more detail. For example, we can see a lower sensitivity of HDAC inhibitors for patients that are pediatric or adult versus, young adult. This is the opposite. We can see, least response again for pediatric, but higher for the other age group for a JAK inhibitor. We can also look at subtype specific responses. Like, is there any drug for which a specific subtype of sarcoma has enhanced or lower sensitivity than average? And so if you take all of the sarcomas we screen and look at osteosarcoma, we can, for example, see a higher response to everolimus, which is an mTOR inhibitor, which is actually used clinically for osteosarcoma. With, there is a huge diversity in there, but on average, they're more sensitive. And conversely, chordoma, which is a very drug-resistant type of bone tumor, tend to be non-responsive to the same drug. We also can see different trends within histology. This is leiomyosarcoma. Topotecan was shown maybe a couple of decades ago by the GOG as non-effective in leiomyosarcoma, and we can see that leiomyosarcoma tend to be least affected by topotecan than any other sarcoma we test. But for example, we see a strong signal of enhanced responses to dovitinib, which is actually now in trials in combination for leiomyosarcoma. I did tell you that there are trends, for example, sorafenib, everolimus. Also, we can see a strong trend for osteosarcoma, which would be this teal color as being the most responsive. But the one thing I want to point out is anywhere where we see a trend, we always have individual cases of patients with the same disease. So these are other osteosarcoma patients here in green, which are actually not responsive at all. And so again, kind of pushing this idea that although we can pick up these trends, there is always some outliers, and we can pick up on this specific outlier. As I mentioned, we also do screening, and I kind of just want to show you a couple of examples of how the functional and molecular profiling, kind of meet. And this is one case of a very young patient with an incredibly aggressive undifferentiated spindle cell tumor. This is now called USS. And these tumors, originally, the primary resection was found to have a PI-K3C alpha alteration, which is associated and actually used clinically also for alpelisib sensitivity. And again, this is a hotspot mutation, and expressing this specific alteration in cell line confers sensitivity to a number of different inhibitors, including alpelisib. So we went in there and we looked at all the different PI3K inhibitor we tested on these patients, and we can see very clearly that all of them were pretty much having a signal of efficacy. When we put the results in the context of all of the sarcoma we tested, in fact, this patient is up here. So very strong sensitivity to alpelisib we see on the organoids as well. We resequence the sample, can find this PI3K alteration there, as we are expecting. Now, very interestingly, just a couple of months after this patient, we got a MPNST patient that was a recurrent. So this patient had a primary disease, which was sequenced and found to have the same exact alteration we found for this patient in the PI3K gene. Now, when the patient came in with these recurrence, we immediately tested this for alpelisib, right? And we found no response at all. So when that happened, we went back, we sequenced this recurrence, and surprisingly, or at least surprisingly to us, we found no more, there is no trace of that alteration left. We don't know why this mutation is not here anymore. We're investigating that. But we could pick up this lack of response that nicely correlated with the lack of the biomarker. Yet, if you look at the sample that had the strongest response we've ever measured to alpelisib, and you look at the DNA of this sample, there is really no rhyme or reason as to why this patient would be responding on the basis of the biomarkers we have now. And so again, the idea is as we collect more and more data, we may be able to make sense of these and come up with more effective biomarkers. There is also, other ways that this type of very rapid screening is actually helpful. And this is just one quick example of a very, very young patient of us. So she's 16-month-old, which had a Thai mass. When you have a patient in this very young age group with a sarcoma, just for the way the disease works, they tend to suspect fibromyosarcoma. Fibromyosarcoma is a disease driven by NTRK fusions, and so typically these can be associated with larotrectinib and entrectinib, any of these NTRK inhibitors that are really successful clinically. So we went in there with a suspect fibromyosarcoma diagnosis, tested the sample. Turns out no response whatsoever to larotrectinib. And a couple of weeks down the line, when the pathology came back in and they looked at the alterations on NTRK, they actually confirmed there was no alteration. And this was ultimately diagnosed as a rhabdomyosarcoma. And so we could see this a week after, right? Rather than waiting about a couple of weeks from surgery. So as we think about moving this type of assays toward the clinics, there are a lot of things we have to take into account. As I mentioned, we choose, but we choose, particularly for precision medicine, not to passage these cells ex vivo. When we make this choice, we are left with a huge limitation, which is we only have as many cells as we get from surgery. So we need to take this into account. And the way in which we go about designing our libraries is we try to customize as much as possible, which is, I think, painful but necessary. We prioritize FDA-approved therapies for the specific indication and for other cancer indications. And we want to also look at therapies which are currently in clinical trials. And so we typically come up with a focus screening over a few different concentrations for compounds which are of high interest. And then we use the remaining of the cells to do larger screening. Those are discovery screening, conventional designs, single concentration, which we look at. And if we find any hit in this larger screening, we can then go back to frozen cells and confirm that specific hits. So we are limited by number of cells, but we can make do with things as little as needle biopsies. And we have done that successfully in a few cases. This is one example where we had a needle biopsy. This was a kid coming to the clinic and undergoing a biopsy, a diagnostic biopsy for osteosarcoma. And this is how the patient sample look like. So again, kind of always want to keep bringing up this idea that these are sometimes not very cellular tumors, so a lot of matrix with scattered cells. And these are the models we developed. We had the ability to test about, I believe, in total 100 different interventions, and we saw a signal of activity against platinum therapy. Now, this patient went on to get neoadjuvant combinatorial therapy that includes platinum, and a couple of months after undergoing resection of the tumor, as you can see, there is basically no tumor cell left. There was over 100% necrosis. And, in osteosarcoma, this very high level of necrosis at time of surgery post neoadjuvant therapy is actually a really good indication of a positive, prognosis and a favorable long-term outcome here. So this patient is very likely to do really, really well. So we knew that ahead of time, right? And that's great. Can we actually do a little bit better and really match this 3-drug regimen called MAP that these patients get? We can do that, and let's see how the organoid response correlates to necrosis, right? So we can see good correlation even of the full 3-drug regimen, where we have cases where the organoids are dying significantly. The patient ended up having a higher % of necrosis than patients where the organoids are actually surviving treatment, and then the percentage of necrosis is much lower. And so this is great, but it is logistically really challenging for us to get these diagnostic biopsies outside the context of a trial. And so we're trying now to build in a small trial where we will get a couple of cores from a diagnostic biopsy and determine the predictive power of this assay in a little bit more of a structured manner. And I think the positive thing here is that there is really no extra procedure or anything additional that the patient would need to do. We just really are going to test this regimen, and this is going to teach us something about the assay. But the hope is in the future we will eventually be able to also use this information clinically, right? Like if you know ahead of time a patient is not going to respond, can you move on to the next, therapy that you could consider and you would consider a few months down the line. So as I said, this patient getting, or any patient with osteosarcoma getting platinum as first-line therapy is absolutely nothing surprising. This is standard of care for these patients. But what I always like to show is this type of picture that shows you all of the NCCN recommended regimen and how many different options there are for second-line therapy for osteosarcoma. So this is disease that is either metastatic or relapsed or refractory. So there are so many different options, right? And no real preponderance of evidence as to which one would you pick and choose to give to the patient. And so the oncologist ends up having to make this decision in some ways blindly, right? And so what we want to do is we want to be able to provide additional information to be able to pick and choose which regimen is better for the given patient on the basis of data we can generate. We are looking more broadly at how well our assay is able to predict clinical response. Again, these things outside the context of a trial are a little bit challenging. The way in which we are looking at these, we are looking at time to the next therapy, which is kind of a real world measure of how well a patient is doing. And so the idea is the patient with advanced disease will keep moving on from therapy to therapy. And as the time passes, as the tumor evolves and progresses, the patient is likely to stay less and less time to each subsequent line of therapy. However, if you give an intervention that is actually working well, what that is likely to do is likely to extend the time that the patient can stay on that specific therapy. And so what we are seeing is that therapies which are really working well on the organoids in the lab also are doing well in the patient, and vice versa. Therapies which are not at all, if at all, actually, inducing growth of the tumor in the lab, also end up for the patient, on the patient side, the patient end up spending very little time on that therapy. And so we can make these correlations. And again, the idea here is now to open this in the context of a trial to have a little bit more control over the over what's happening and the type of intervention. But the other thing we are looking at is, well, okay, say this type of assays, moves forward and is able to be used clinically to pick and choose interventions. What are the drugs that are coming up from this screening, and are these predictions actually actionable at all? And so I don't think anyone has looked at these in the context of this many, tumors, etc. But that's what we looked at. And so we looked at what are the most effective regimens for each and every patient, and we capped that at 5. So we're very strict with the number of drugs we're looking at. Not only that, but we cross-reference these with the pan-sarcoma data. And so we only consider an intervention if it is between first or among the first 5 top most effective drugs for each patient, and that patient is amongst the top quartile of responders across all of the patients we screen. And what you end up seeing, and this is just a very small, snippet of a much longer list, but we are seeing a lot of interventions which are actually approved. The most part, we are seeing FDA drugs coming up, which are approved, but for a different cancer. With, some exceptions, sometimes we are seeing drugs which are approved for the specific cancer type we're looking at. On average, we are finding that with these criteria of inclusion, we find at least one FDA-approved or NCCN recommended regimen for close to 60% of patients. So a really, high percentage of cases where we find some drug that is not just the tool compound we use in the lab, etc., but is actually a drug that could be potentially given to a patient with some breakdown shown here. One thing we also get asked a lot is, how does your assay compare to PDXs? As I mentioned at the beginning, a lot of the sarcoma types we study cannot be grown as PDXs, but some can. And this is just one specific example where we can do screening in the organoid side, we can do screening in the PDXs, and then we can try to correlate and see how much or how well do the responses in these 2 models match. And you have to take into account that this is done within a week of surgery, and this is done within 6 to 8 months. So lots of time passes, lots of things are happening to this tumor. Regardless, we actually see, decent correlation between responses. And so that is very encouraging, obviously. Now, as I have said, this is all great. We can start to design these trials to prove the clinical efficacy. There are other issues, however. There are regulatory issues, in terms of really, clinical regulatory issues. There are obvious issues to access drugs. But the one thing which I do want to bring up, and, as anyone thinking about using literally any type of assays that samples a tumor to determine a drug, needs to take into account tumor heterogeneity, which is really linked to also sampling bias. And I'm going to go into this in detail in a minute. And so the idea is there is a lot of heterogeneity here, which we need to be mindful of. And one quick example is this, and I like to show this because chordoma, as I mentioned earlier, this is a very rare malignant bone tumor coming from embryonic remnants of the notochord. It's a 1 in a million people diagnosis. And these patients are typically not treated with chemotherapy. This is considered a fully chemo-resistant tumor, very indolent, slow growing. Patients are treated surgically. We had a very young patient, so a patient in their 20s, with a very aggressive chordoma that was metastatic. And we had the ability to get 3 different samples over the span of 11 months. And you don't have to look at the detail, but what I just want to point out is that even though these samples, as you can see, they're really indistinguishable for all means and purposes, when we drug test them, we actually see specific patterns. We see sensitivities that goes away. We even see the opposite. We see different, sensitivities arising over time. And we see, specific patterns like these. Now, this is very interesting because omeprazole is an mTOR inhibitor. And we saw sensitivity on the first sample. Then the patient actually went on a completely different mTOR inhibitor. And what's interesting is the sample procured immediately after that lost that sensitivity. But then we, for some reason, find it coming back after some time. And so again, a lot of differences here and things that happen. And if you sample a single one of these tumors and you decide on an intervention, it's not for sure that that intervention will work on the remaining of the disease that is, in the patient. So we're trying to study this systematically. We're trying to develop this patient series where we can have a biopsy, we can have a resection, we can have individual metastasectomies, and we can develop organoids for all these different stages. We know what's happening with the patient, and we can try to really see how different they are. And so for cases particularly where you have multiple tumors present at the same time, and we can study them individually, we can match this to how they grew in the patient, we can match this to how they grow in vitro. And again, just a very small portion of this one metastatic osteosarcoma patient where we had individual samples, seeing really the same thing. You have different patterns of drug sensitivities, and metastasis to metastasis or tumor site to tumor site, you see a huge difference. We are coupling all of these in collaboration. For example, in this case with the Boutros lab, we are trying to look at, what's happening in terms of the genomics of this disease, which type of alteration are changing. We are looking at subclonal distribution. And one thing I always, want to point out is, as we look at this to understand and decipher drug response, we are also sequencing both the patient sample and the organoids. And I think what you can see is that everywhere where you have a pair of tumor and organoid, you see how well these tumor organoids we develop are at maintaining even the integrity of the tumor down to the subclonal population proportions. And this is really validating the models we develop, right? Showed you a few examples of heterogeneity in bone sarcoma. This is actually also true in soft tissue sarcoma. Again, one of our very, very young patients with epithelioid sarcoma. This was an epithelioid undifferentiated sarcomas, and these are 4 tumors that were literally just centimeters apart, which we got as, during an amputation surgery, a palliative amputation. And again, you can see how different these tumors, which are very, very physically close, all present at the same time, are in terms of responses. And so say if you only get tumor 1 and you perform a screening of that, and you choose a drug to give this patient, but the majority of the disease looks like tumor 2, your intervention is not likely to work. So we need to think about these problems, and we need to think about how to deploy this type of technology in a way that makes sense. One way we are trying to investigate these heterogeneity better is by looking at single individual organoids and what is their response to therapy. We are able to do that now because we have a modified platform where we can couple bioprinting, and so we can generate very thin matrigel constructs with organoids growing in them, which is amenable to new imaging capabilities. And so together with the Title Lab, in this case, we are deploying high-speed live cell interferometry, which is a fancy way to say we're measuring the weight of organoids and how does that change over treatment. So bioprinting has been really transformative for us because instead of having to generate all these different rings by hand, we can now have a machine to do that for us automatically, which has been, again, great and really opens up all these different opportunities. This is how the live cell interferometry images look like. So you can see the individual organoids growing in culture and how they change over time. And, darker means heavier and lighter means less weight. And so you can see how they distribute weight over time, etc. And so now we have these imaging capability where coupling, with machine learning tools, we are able to have spatially and time resolved, label-free mass measurement of thousands of organoids at the time. And what this is allowing us to do is really to pick up these possibly resistant clones and these heterogeneity. We can pick up heterogeneity even within very well-established cell lines, right? And so here is just a quick example where we have 2 breast cancer cell lines. one with a high overexpression of HER2, and so should be responding to HER2 targeted therapy, and the other one that doesn't. And we can definitely see more and more organoids losing weight, and so losing weight in a non-healthy way, so dying off as you treat them with an HER2 targeting agent. But we also have the ability to look at the proportion of organoids, which actually are not only maintaining their weight, but some of them even still gaining weight upon therapy. And we can go back into the images, and we can find these individual organoids that show this very different heterogeneous pattern of response. And the idea now is to be able to pluck these out and study them individually. And this is not theoretical, this is not just cell lines. We can actually apply this type of platform also to sarcoma, to do larger drug screening and identifying responses in this way. So this is just one of the things we're doing with the platform. To conclude, I also want to mention that we have different type of expansions to our pipeline. We have, for example, mini liver inserts, where we try to put a liver just where we need to metabolize a drug that is given to patients as a prodrug. And so you wouldn't be able to test this in vitro, you could only do this in mouse. But if we have a liver where we need it, we may be able to test this drug and metabolize them where we need them. We have co-culture model with vasculature we're developing. We have an immuno-oncology platform we're working on. We have a hypoxia platform we're optimizing, and a good-to-go chemoradiation platform to study both the drug plus radiation, which is, again, very important for tumors like sarcomas or soft tissue sarcoma in particular. So this is all optimized as we have it now. We have, a simple platform, automated, few cells needed, no passaging, and screening of highly personalized models within a week from surgery. And we can use this for personalized prediction. We can use this for, developing prognostic markers. We can use this as a diagnostic tool, as I showed you, and to really perform this broad landscape analysis and characterize heterogeneity. So this is what we've been up to over the past 3 years, and none of this would be possible without the work of these wonderful people in the lab. What I showed you today in particular is the work of Quinn, Peyton, Hamad, and Bowen here. We have a terrific group of collaborators that work with us, in particularly on the science side, the Boutros Untitled Lab, as I mentioned, but all of our clinical collaborators. Nothing that we do would be possible without them or without the support of our funders, and in particular the NCI, also the Cancer Moonshot and the Alex Lemonade Foundation, which have been supporting our sarcoma work. And I just want to say we're hiring at many different levels. And so, reach out if you're interested in rare cancers or in organoid models of disease. And thank you, everyone. And I would be happy to answer any question if there is any.
Thank you very much. I love the plug for hiring. That's great.
I mean, right?
Yeah. We have some questions that came in. One of them you actually answered. They sent an update and said they got their answer, but we're going to start. We're going to jump right in with questions. If you have questions, click on the Q&A button on the bottom of the screen. You can submit your question there. So the first question says, have you looked at combination therapies yet? It would be very valuable to exhaustively annotate drug synergies.
Yeah, we do. And, we have the flexibility of looking at combinations in a few different ways. The combination I showed you earlier, which are combination done, like MAP or sorafenib everolimus, so combination which are given clinically. We typically do that by adding the drugs at the same time, but we also have the flexibility of, for example, of testing, sequential treatment, etc. So we do that. Absolutely, yes. And it's very important, I agree.
Okay, the next question is from Chris Sorosiak. Says, great talk, Alice. I'm wondering if you incorporate any consideration of toxicities in the therapy assignment that is produced by the organoid screenings. We know that certain drugs are more toxic in pediatric patients than adults, like doxorubicin, causing cardiotoxicity at a much higher rate. Are you able to take this into account when making recommendations for treatment?
So this is a great point. Again, we are not at the stage where we are using this to actively guide treatment, but I think this is a absolute important point and definitely something that will be factored into account as we make these recommendations. You may be familiar with, there are some screening types where people take normal cells and do screening on normal cells at the same time. Personally, I think that's very valuable if you have the normal counterpart from the same patient, not as much if you're taking just, cells that grow in the lab from a completely different person. We do that whenever we can. So we get the normal tissue and the tumor tissue, screen both at the same time and look for like patient-specific, possible toxicities there. This is very challenging in sarcoma. So if you have a kidney tumor, right, they take out the whole kidney, you have a piece of normal kidney, piece of kidney tumor, very easy to screen both. For sarcomas, it's even challenging to know what the tissue of origin really is sometimes. So we cannot really do that in a targeted manner. But I think these are absolute, the idea of taking into account toxicity, it's really important and definitely something we should be doing once we start guiding therapy.
Okay, next question says, great work, Alice. One question, how do you address heterogeneity in multifocal disease?
So, that's exactly what we are trying to do. So we are trying to, this is obviously challenging unless the patient is undergoing specific types of surgery, right? But whenever we can get individual, tumor, individual location, that is exactly what we are trying to do. Of course, this is not going to be feasible for every type of patient, right? But the idea is, can we learn something about the biology here? Can we, that will allow us eventually to extrapolate what we learn from the patients where we can get individual tumors, more broadly. The other thing we're looking at, and I didn't show that today, but we are trying whenever possible, not only to look at different, locations within a patient, but also when you have a larger tumor, how heterogeneous is that? And so large primary tumor, we're trying to take different parts and develop organoid individually and look at, for sequencing, but also drug sensitivity individually. Again, with the idea that you go in, for example, with a core needle biopsy, you're sampling teeny tiny part of a potentially much larger tumor. How representative that is in sarcoma. And we have good answer for other tumor types, but I don't think we have enough yet on sarcoma to know the answer to that.
Okay, next question is from John Bushweller. It says, are you open to testing novel agents that are at earlier stages of development?
Oh, absolutely. Yeah, I think, the idea of this type of approach is really to potentially help some drugs going to market. You have to think some drugs which have on a completely unstratified patient population may have 5-10% response rates. Those can be good responses, but that proportion is so little that these drugs have a completely impossible path ahead to get approved. And so if you have the ability to do this in the context of like companion diagnostic, help refine the patient population better, I think something like this can really help some drugs moving forward and find potential indications. So we can definitely do that with early stage compounds, yeah.
So I guess that means that this person could get in touch with you.
I mean, sure, if they have a drug of interest, then absolutely. We collaborate broadly as we can. That's okay. I think that's the point, right? And I mean, I guess, and I want to say that, yes, if anyone is interested in this rare cancer, you have a molecule or a pathway of interest and you don't have the models, we have the models and we want to collaborate as broadly as possible because not everyone has access to these rare tumors.
Okay, great. The last question that I see is from Fred Barr. Hello, Fred. Nice to see you here. Thanks for the seminar. The question is, to what extent do these organoids maintain the chromatin, DNA methylation, and gene expression patterns of the tumors from which they are derived? And does that change with time and culture?
Yeah, so as I mentioned, we don't do that for every tumor, but for some tumors, we are doing, passaging and growth in culture over time. And then we go back and we look very deeply at the genome and we look at the gene expression as well. We have looked at DNA methylation of tumors in culture. We've not done DNA methylation for sarcoma, to be honest, but we've done that for other tumors. And I think the simplified version of the answer is within our unpassaged organoid, so the 1 week in culture, as you can imagine, not much is changing. We see really consistent, really consistent DNA, really consistent methylation for what we looked at, the really consistent RNA expression profiles. I think that's intuitive because they stay such a short time in culture, right? As we keep passaging, things, keep happening. The one thing I wanted to mention is we don't do any selection or any sorting of the cells. So anything which is in the tumor, we put in culture. And so there is a component of tumor cells. There is also a component of, support cells and microenvironment cells of different types. And we think that's fundamental. There is lots of data showing how that influences drug responses. We want those cells there. We want the proportion of that cells to stay the same. However, one of the things we see happening as we passage these cells, we see, for example, that you're purifying the tumor, right? And so these are things that do happen with time in culture. You end up having mostly, in the majority of the systems we looked at, you end up having more tumor cells, less microenvironment component. And the other thing we have seen is that there are, of course, changes in RNA expression, and there are changes in drug responses, which typically are reduced sensitivity over passage. So you end up with models that are more and more resistant.